Welcome to a short introduction to the Ontology Search in the BKL, the knowledge library that contains the databases TRANSFAC, HumanPSD, and TRANSPATH. The Ontology Search is a powerful tool that enables searching for data types using curated and calculated properties. Search and combine categories for 4 data types: genes, including protein properties, microRNAs, diseases, and drugs. To start, open the tool via the main search menu in the upper left corner. The interface is composed of 3 sections: the query builder in the upper part, the vocabulary driller section below, and the lower part where the result will appear. You can select 4 searchable data types in this drop-down list. For each selection, the respective properties will be loaded into the driller section. The numbers in front of each category depict the number of entries, here genes, associated with the property term and its children. To drill into the ontologies, select a more specific term in the rightmost pane. If it has a greater than symbol to its right, it has children that will be displayed in the next pane. Another way to open the ontology search is from a search result or uploading a list of entries. The resulting entries can be sent to the ontology search by clicking the link above the result. Once the loading of the Ontology Search interface has finished, the numbers of associated entries in the driller section have adapted and filtered for the input that was sent to it. In the first example, we will identify post-translationally modified transcription factors as targets in various signal transduction pathways, which are also biomarkers for certain diseases. To find the category that restricts for post-translational modifications, I'm using the category search. The results can be selected from the drop-down menu to the right. Explanations regarding the prefixes are given in the nearby link. I'm selecting the best matching category and it appears in the query builder. To apply this filter, I'm clicking the search button there. With the next condition, I'm narrowing it down to human genes only. Again, with the category search, I'm looking to restrict for transcription factors. While adding one search condition after another, see how the number of associated genes decrease in the driller as we refine the search. Next, I'm selecting those genes involved in signaling pathways. The genes I'm looking for should also be biomarkers connected with certain types of cancer. After selecting liver neoplasms, I'm adding an OR operator to add a second condition, this time gallbladder neoplasms. With all specifications set now, the resulting genes can be retrieved by clicking on the View All 227 Genes button. Scrolling down, there's a result list of genes. I can select some or all and have several options to proceed from here. I could send the genes to the match suite to analyze how these genes are regulated. But now I'm more interested in finding out which signaling pathways are involved and how they look like. For that, I'm sending the result as input to another search by selecting Pathway from the drop-down menu. The result of this second search are signal transduction pathways that have participating genes and proteins from the first result. To view a pathway, select it and send it to the Pathfinder tool. The Pathfinder displays the components of the pathway in their intracellular location. In a second example query, we want to find transcription factors which are expressed in certain tissues and relevant for specific diseases and their positional weight matrices to be able to create a tissue-specific matrix profile to be used in binding site prediction with the match tool. First, I'm selecting all transcription factors that have a binding site matrix. Then, again, I'm focusing on human genes. Next, I'm narrowing the set down to factors expressed in specific tissues. I'm selecting liver and gallbladder, using the Add OR Terms link to add an OR condition. To add relevance for diseases in liver, I'm adding liver neoplasms as next search condition. Now, there are 265 human transcription factor genes in the set to be retrieved. To obtain the corresponding matrices for this gene result set, I'm using the Search Within result options. This time it's TF binding matrix. For many transcription factors, several matrices exist in TRANSFAC. To focus the matrix set, I'm applying the filter tool. Only matrices with the keyword recommended in the type field should now be selected. With all resulting matrices marked, I'm sending them to the Profile Creation tool. Here you can define different kinds of thresholds for the matrices, but we are going with the defaults. After providing our profile with a name, it can be saved. To apply it in a test run of Match, click on the link. Here, we are using an example promoter sequence to predict binding sites with our liver cancer specific profile. In the match result, all matrices from the profile with the binding site hit on the example sequence are listed and the predicted sites are visualized. Thanks for watching. If there are questions, you can always contact us at support@geneXplain.com.